All right, so we're going to focus in on, as I said, the surface to volume ratio of a cell. Not just prokaryotic, eukaryotic, but we're going to get to that to be really specific about it. But first off, we'll just talk about increasing size. So let's just say these two little cubes here, the cells aren't cubes, but it's just going to make it a lot easier for us to talk about dimensions and have for you to kind of see the dimensions if we use a cube shape for sides, of the cube and assign numbers to them. All right. Now, when we talk about prokaryotic and eukaryotic cells, we say the prokaryotic are small and they're usually limited in size, and then the eukaryotic are large and they can tend to be much more variable in size and get incredibly, incredibly large. And the question again we're saying is, how can they do that? And I'm telling you the answer, it has to do with, it's not just a simple answer, but it has to do with surface to volume ratio. So what the heck are we talking about with surface to volume ratio? We're talking about surface of membranes. So cells are made up of a membrane, and then there's an internal environment, a cellular environment, and there's an extracellular environment, everything that's outside the cell. So the membrane is what separates those two environments, inside the cell from outside the cell. That's the membrane, right? And so if we measure the area of the surface, that's the surface area of the cell, okay? And then the volume is everything inside the cell. So volume and then surface. Now, when we talk about prokaryotic and eukaryotic, one of the first things that typically comes up beyond size is nucleus. People say, oh, nucleus. The prokaryotes don't have a nucleus. The eukaryotes do have a nucleus. Okay, so this is, say, representing the nucleus. And that's made up of a membrane. What you will find is that the nuclear envelope, and this is future for this particular course, is really made up of two membranes, two membranous layers. So there's an inner membrane and then there's an outer membrane. And the outer membrane then creates this group of highly folded sacs like this, called, which you may have heard of this and you may not, the endoplasmic reticulum. Okay, I'm just going to try and give you a little idea here of some of this. So now this structure here, label it just ER. Don't worry about this right now. It doesn't have anything to do with this specific lecture. I'm just using it as an example. It's a whole series of membranes that are all folded throughout the internal environment of a eukaryotic cell. So what we're seeing is that's all added in. So that's part of surface area too. Okay, so the surface area we're talking about is not only the outer surface area, but it's all membrane surfaces. So that's the outer membrane plus anything that happens to be inside. Now for the prokaryotic cells, our example here, let's say a prokaryotic, they have no internal membranes. And if you take a microbiology course, you'll find that's not, it's basically true in the sense of what we define as a membrane and all the proteins associated with membranes and so forth. However, prokaryotic cells are not just a little sac full of enzymes and other sorts of things. They do have sort of some single layered compartments, there are granules, there are inclusions, there's all sorts of other things that allow them to be a little more complex than they're usually the way they're portrayed. Okay, but overall, we don't typically have these types of membranes and many others, because eukaryotes are going to have, this isn't even it, there's tons of other sorts of things that we're going to have inside the eukaryotic cell. So let's start to talk about this surface to volume and then the ratio. That's just the proportion of one to the other. And then we'll get at why that's actually important. So let's say these are two cells and they happen to be cubes. All right. And this one is 1 centimeter, so that's the height, width, and so forth. And this one is 5 centimeters. And you might see this example in other places, but I'm going to really try to break it down for you and do the calculations so you can actually grasp what we're really talking about here. Okay. So if we calculate surface area, for surface area we're going to get the height times the width, like that, then times the number of sides, and then the number of, now let's say cells, or cubes. We've got cubes. So the height is, in this particular case, 1. The width is 1. The number of sides, well, it's a cube. Okay, so it's got a front and a back, 1, 2, and then 3, 4, 5, 6. If you go around it, 6 sides. And in this particular case, we only have one. We just have the one cube. So in this example, for this particular little cube here, if this was our cell, our 1 centimeter cell, let's say, its surface area is 6 in terms of all the sides for the 6 cubic centimeters there. Now, the volume is going to be similar, but a little bit different. So here we have height times width times length, the 3 different dimensions instead of sides, and then the number of cubes. So for that, we're going to have height is 1, width is 1, length is 1, number of cubes is 1. All right, so that's going to be 1 cubic centimeter for volume. So the ratio for this little cube here is 6 for surface area, and volume is 1, so 6. Again, don't worry about what it means right now. We'll get to what it means. We'll look at the comparison. Here's this bigger cube. So let's imagine a cell. We said prokaryotic cells can change their size. They're not all the exact same size. Some are bigger than others. And none of them have membranes inside, really, or a nucleus, except we talked about that new discovery, strange example, which is something that's going to maybe change even the way things are defined in the future. Who knows? But for now, this is kind of what we're working off of, our traditional definitions. So now for this particular cube, we're going to change some of these things. We have our height as being 5 and our width as being 5. The number of sides are still 6, and so we only have 1 cube, right? So we have 5 times 5 times 6, which is 150. So for this, we've got now 150. And then for the volume, we're going to have height times width times length. So it's actually going to be a pretty similar number here in terms of the calculation, 5 times 5 times 5, which is 125. So for this cube, surface area 150 to volume 125, we see a much smaller sort of ratio versus 6. What does that mean? Okay, so in terms of numbers, big to small. So essentially here it means that there's 6 times the surface area compared to the volume. And if this cell simply got larger, what it tells us is that it's still not a proportionate 6 to 1, right? Now the surface area and the volume are similar, which means the volume has gotten larger and the surface area has gotten larger, but they haven't increased at the same rate. So this is an important thing. Remember, surface area and volume do not increase at the same rate. Volume, just to simplify it, more. Volume's going to increase more than the surface area will if you just get bigger. Again, why does that matter? So what? Well, the surface of the cell is where often there's going to be exchange of everything. So say, oxygen coming in, carbon dioxide coming out, nutrients coming in, other waste going out of the cell. The membrane is not just a static barrier. The membrane is a site of enormous activity. Right? There are all sorts of proteins associated with the membrane, and they're moving things in different directions. Now, some of these things, it's regulating the natural movement of them from high to low concentration. Other things, it's pumping and pushing. It's pushing things against the way they would normally flow. So say like a ball would normally roll downhill, that would be natural. So in cells, things are going to move from high to low concentration. That would be natural. But cells can then push things the opposite way. They can push things from low to high concentration using energy. Right? The same as you can push a ball up a hill. It's not going to roll by itself, but it could be energy can be used to do that. So cells are going to bring in extra nutrients. Even if they already have nutrients, they're going to bring in more nutrients, and it's going to require energy to do it, but they'll get more out of it. So all those activities and actions are going to take place at the surface of membranes. And the more membrane you have, the greater the rate of these sorts of processes, getting nutrients and oxygen, getting rid of waste. The less surface area you have, but a bigger space, start to come with problems. Right? Now, you need more oxygen, but the amount you're getting across that surface maybe isn't supplying you with your demand. Right? You're accumulating waste inside the cell, and you have to get rid of it, but it's taking longer to get rid of the waste because you have a lot more volume, a lot more space. So let's say this is a waste molecule. Okay, so that little dot there, that's a waste molecule, and it's just gonna move out of the cell. Okay, so it's just gonna go out of the cell. And here it's gonna go out of the cell. Just by looking at this, the time it would take for this molecule just to move on its own out of the cell versus this one to move on its own out of the cell. Is it gonna be the same? No, it's gonna take longer because this cell is bigger. All right. So it's gonna take longer to remove waste molecules. More waste molecules will then accumulate. The concentration of waste will accumulate, and cells could potentially poison themselves because they kind of get too big to control all the processes they need to. And that's one of the main reasons why prokaryotic cells tend to be limited in size, because they can't get enough nutrients and they can't kind of get rid of all the waste that they're producing, and they starve/poison themselves after a certain point. Right? That's generally a limiting factor. So that other organism I've talked about in a previous lecture, Thiomargarita, has to be doing something different maybe to get to be so big without all these extra internal membranes. Maybe it has more active transport pumps on the membrane surface or something else it's doing to kind of regulate those processes. But that's the idea. You have to regulate these processes, and all those processes occur at the surface of the membranes. The more membrane you have, the better you can do that. So how do eukaryotic cells get away with then being gigantic, like so big? Wouldn't they then just have the same problem and the problem just be much worse? Well, you have to remember, eukaryotic cells divide up then the cytoplasm, instead of just being one big space like this, into all sorts of little compartments because there's all these membranes. So let's say in this particular cell, let's say it was a eukaryotic cell. It might have a structure inside it, a little membranous ball, a vesicle, that stores waste. So in this particular case now, let's say, instead of the waste actually having to go out of the cell, it's just pulled into a waste collection compartment, right, in the cell, a little vesicle. So maybe it's even quicker. It's taken care of. It's out of the cytoplasm. It's still in the cell, but the cell's going to destroy it, maybe even recycles the material in it so it can reuse it for other processes. It gets carbon from it or oxygen from it. It gets maybe individual elements or pieces of a larger waste molecule it can use or convert into something else, nitrogen, nitrates, and different sorts of molecules like that. So that's what we get here. This is the same sized cube, so this is going to be the 5 centimeters. Right, in both dimensions. Okay, this dimension here, 5. This dimension here, 5. But what's happened in this example is we've broken it up into 5 individual 1 centimeter, so these are now 1 centimeter across, 1 centimeter tall cubes, deep and so forth. They're all 1 by 1 by 1 by 1. So if we go back and do some of these calculations, we say, so this cube and this cube are pretty much the same size. The difference here is this one is broken up into little pieces and compartments. the way a eukaryotic cell. So now this one here is the example for our eukaryotic cell. Remember, eukaryotic cells aren't cube-shaped and they're not broken into little tiny cubes. There's membranous compartments, there's little vesicles, there's mitochondria, there's endoplasmic reticulum, Golgi apparatus, usually round or oval or sort of this amorphous sort of shape, all different kinds of shapes in terms of the structures. But this is just for our example so we can calculate it and put the numbers, in here. So let's go with the eukaryotic one. I'll use this different color so we can separate it. So for our surface area, okay, so we're going to have the same calculation, the height times the width times the number of sides times the number of cubes. So now we're looking at, individual cubes, not the whole thing, right? So we got stuff 1 times the number of sides, 6. That's what we had before. But now, wait, number of cubes is not just 1. It's 1, 2, 3, 4, 5 down, 5 across. So that's 25. And then we have 1, 2, 3, 4, 5 rows of 25. Hopefully that makes sense to you, right? So if we're talking about three-dimensional cubes, that's one cube stacked on top of another cube, on top of another, on top of another. So that's 5 cubes tall. That's 5 cubes tall, 5 cubes tall, 5 cubes tall, 5 cubes tall. Right? So that's just one row alone would be 25 cubes. We have 1, 2, 3, 4, 5 rows of cubes. Okay, so that's 125 individual compartments. See, and this is kind of the idea we're getting out the eukaryotic cells. Compartments, lots of compartments. So what do we have here? So now we have 1 times 1 times 6 times 125. That's 750, right, for the surface area. Now what about volume? Volume's going to be big still, right? But here we've got volume, if you remember, is just height times width times length. So each individual cube is still just 1, 1, 1, 1. But then it's the number of cubes. And the same as here, we have 125 cubes. So now volume is going to be 125. So what do we have here for the eukaryotic one? This, in our sample, we have 750 for surface area over 125 for volume. You divide these two and you get surface to volume ratio of 6. Essentially the same as we had up here. So what we have is a structure that's 5 times larger, okay, than this original little cell. However, the efficiency of the cell, its ability to do everything, transport waste and nutrients and oxygen and carbon dioxide and store things and all that, is equal to something 5 times smaller because it's able to break itself up into little compartments, okay, by having internal membranes. So the key to surface to volume ratio and eukaryotic cells is eukaryotic cells have internal membranes. Internal membranes increase the surface area. Increased surface area increases efficiency of the cells. Efficiency for what? For everything. For everything that the cells do, they can do it better because most of the actions and activities are going to take place on the surface or across a membrane. Membranes are not, again, going to be a static barrier like a plastic Ziploc bag containing water inside it. That's not what a cell's like. That membrane, that barrier is dynamic, right? It changes shape. It has proteins associated with it. They change shape. They move things across it. They regulate what goes across it. They can set charges different from one environment to another. One environment could be positively charged. Another environment could be negatively charged. All different types of molecules can be sorted into different areas. You could have nutrients here and then waste over there, but they're all separate from one another. You could have enzymes that digest things that could destroy everything in the cell, but they're kept in a compartment, a little compartment that then is just used to digest all these waste molecules and cut them up. So having internal membranes is the key, and internal membranes then comes related to our definition, right? Prokaryotic, eukaryotic, we're talking about nucleus. Nucleus is a membrane. The real difference between the prokaryotic and eukaryotic cells is having the internal membranes. Right? Prokaryotic cells typically don't have the true membrane structure. Now, in my intro cell biology, general biology class, the entire second exam is all on membranes. Right? It's because they're so important that everything that really happens in the cell happens at the surface of the membrane. It's very important you understand structure of membranes and all the actions and activities that take place at the surface of a membrane. So that's kind of what we'll spend a good amount of time on that. So hopefully this gets you past this concept, all right, of this topic of the difference between pro and eukaryotic cells, the size difference, and the explanation of how can eukaryotes be so much bigger and how be so much variable in size. They could be little, they only need to be small. Big isn't necessarily better in this particular case, it's just efficiency is better. So doing the job that you need to do well is the goal. Right? And so by having more membranes, you can do that, regardless of your size, small or large. So hopefully that will clear up this for you. We aren't going to be running calculations for students in the class, but if you look at these calculations and maybe compare them to some other images you find in the textbook, hopefully this explains it, and you can kind of see what I'm talking about.